So we're going to finish up our lecture talking about DNA replication. And in class, we left off talking about the structure of DNA. And we talked about how the building blocks or the monomers of DNA are going to be your nucleotides. And remember that a nucleotide has three parts. It has a phosphate, a 5-carbon sugar, and a nitrogenous base. And that the building blocks of DNA get put together to make the polymers, which are the nucleic acids. And if you look at RNA, remember that we talked about that RNA is single-stranded, while DNA is double-stranded. And if you look at a double-stranded DNA molecule, you'll notice it kind of resembles a ladder. It has a backbone, and it has rungs from the ladder. And the backbone, remember, are the alternating sugar phosphates. So there's sugar, phosphate going down the strand. And that's on both sides of the ladder. The rungs of the ladder, remember, are going to be your nitrogenous bases. And we talked about that for our base pairing, A is going to pair with T and G is going to pair with C. And so remember that we said that the reason that DNA pairs the way that it does is, remember, purines, shorter name, larger ring structure. Purines are 2 rings big, while pyrimidines, which have a longer name but a smaller ring, pyrimidines are smaller rings. And notice that if you put 2 purines together, that would be too wide. If you put 2 pyrimidines together, that would be too narrow. However, if you match a purine with a pyrimidine, and that's the same going all the way down the strand of DNA, what you're going to get is a DNA double helix with uniform diameter. And this was consistent with the X-ray data from Rosalind Franklin. And so this is just another image of the structure of DNA. Okay, again, remember I said it kind of resembles a ladder, except that this ladder is actually twisting, and that's what we call the double helix. This is just a couple diagrams of different structural models of DNA. On the far left, you can see the ribbon model, and in this particular model, you don't see these sugar phosphates, just like notations of them, so blue, yellow, but you can see the base pairing, so you can see G pairs with C, A pairs with T. And remember that we said that the bonds between the base pairs, those are hydrogen bonds. And you're going to see in a minute that that's important because that weak interaction between those bases allows for the 2 DNA strands to separate during replication. The structure in the middle, this is a partial chemical structure. It shows you some of the chemicals that make up DNA. So you can see the phosphate, you can see the 5-carbon sugar, you can see these nitrogenous bases, but it doesn't show you all of the atoms on the DNA molecule. And then the far right is just a computer space filling model, just showing you the overall structure of DNA. So we mentioned that the 2 strands of DNA run anti-parallel. And what that means, remember, is that one strand runs 5' to 3', and the 5' is going to be, if you look at the sugar's numbered 1, 2, 3, 4, 5. It's a 5-carbon sugar, remember. And those 5 carbons are numbered, and they're given a prime designation. That's that little line there. So the 5' end is going to be the free 5' phosphate. Notice if you look at the rest of the 5' phosphates along the strand, it's participating in that covalent bond that's joining the nucleotides together. So this one's not free, this one's not free, but the only free one is going to be at this end, and we call that particular end the 5' end. Now conversely, on the 3' carbon, normally you'll have these OH groups, these hydroxyl groups. Notice here, the hydroxyl group is participating in this bond. Same thing here and here, but this is the only free 3' hydroxyl group. And so we refer to this as the 3' end. And so if you look at the 2 strands of DNA, this one goes 5' to 3', and notice that the complementary strand, notice up here is the 3' end. So here's that hydroxyl group that's free, and on the strand on the bottom, there's your 5' end. And so notice that these 2 strands run in opposite directions. One goes 5 to 3, the other goes 3 to 5, and we call that orientation anti-parallel. Okay, so anti-parallel means that they run in opposite directions. So, question for you, and I would like you to pause the video and think about the question, and then when you're ready, go ahead and turn it back on. I think we already talked about this one in class, but it's a good review. So, what type of bond joins the bases of complementary DNA strands? Okay, so go ahead, take a minute, pause it. So if you said hydrogen bonding, you are correct. Okay, and so the hydrogen bond is going to be the one that joins complementary DNA strands. If we were talking about the backbone joining nucleotides together, that then would be a covalent bond. But in this particular instance, the answer is a hydrogen bond. So let's talk now about DNA replication and how it occurs. When looking at DNA replication is semi-conservative. And what that means is that when you look at an original parental DNA molecule, okay, so here is our parental DNA, and remember that before mitosis occurs, DNA needs to replicate. It needs to make an exact copy. And so that parental strand of DNA is going to separate, okay, and so the two strands are going to separate. And remember, that's why those hydrogen bonds between base pairs are so important, because that weak interaction allows for the two strands to separate. And so the two strands are going to separate from one another, and those template strands act as a template, and DNA polymerase, an enzyme, is going to come in and it's going to read the sequence of bases. So where it sees an A, it's going to put in the complementary T. And where it puts in a C, it's going to put in a G. And when it reads T, it's going to put in an A. And it's going to do that on both template strands. Okay, and so both of the parental strands act as a template to form a new DNA molecule. And so what you get when DNA replication is finished is that you get two DNA molecules, and each double helix is made up of one original parental strand, okay, that's the darker blue, and one newly synthesized DNA molecule, that's the lighter blue that you see. And so this is semi-conservative. You're conserving the parental, but you're also adding a new strand to it. And so this is just a diagram showing you semi-conservative replication. And in this particular diagram, the blue one is going to be the parental strand, and notice that it's opening up, and as it opens up, a new complementary strand is synthesized by reading the template strand. Now, replication begins at origins of replications. These are specific sequences on the DNA that are recognized by specific proteins. And when these proteins recognize these sequences, they're going to unwind the DNA. And so they're going to unwind it, and when they do that, they're going to form this replication bubble. Okay, and so they're going to separate the two parental strands, and once they do that, now replication can begin. And it's important to note that if you look along a chromosome, each chromosome doesn't have just one origin of replication. It's not that it's going to start at one end and go all the way down in one motion. Instead, if you look along a chromosome, you're going to see multiple origins of replications. And so all of these replication forks, or all of these replication bubbles, are going to unwind and separate simultaneously, and the new strands are going to be made in both directions simultaneously. And so what you get is something called a replication fork. Okay, and so the DNA replication is going to occur. It's going to go into the replication fork until it makes the new strands, and those new strands fuse with one another. And now you get these two daughter DNA molecules, each one having a parental strand and a new daughter strand. And on the right here, this is just showing an electron micrograph, just demonstrating this actually in vitro. And so you can see here's our double strand. Notice here, this is our replication bubble. And so the arrows denote the replication forks, that's the direction replication is occurring. And then down here, here's another replication bubble, and then again, another one down here. And so this just shows you that this happens simultaneously along the length of a DNA molecule. Now, many important enzymes are involved in DNA replication. Helicase, ACE tells you enzyme, helicase is responsible for unwinding the double-stranded DNA. It's the one that's going to separate the two strands. DNA polymerase, polymerize means to synthesize, okay? So DNA polymerase is the enzyme that polymerizes or makes DNA. And so it's going to synthesize and add nucleotides to the new growing strand of DNA. Now, one of the important things to note about DNA polymerase is that this enzyme can only go in the 5' to 3' direction, meaning that it can only add on new nucleotides to the 3' hydroxyl group. And this is going to be important in a minute when I show you a diagram of how DNA replicates. Another important feature of DNA polymerase is that it helps with editing out mistakes. Basically, it's a proofreading enzyme. It's going to recognize if it makes a mistake during replication and hopefully repair it. And if it doesn't repair it, that's when you get mutations. And we'll talk a lot more about mutations later on. DNA ligase, ligase refers to ligate or to put it together. DNA ligase joins small fragments into one continuous chain. And you'll see this again in a minute. So let's take a look at how DNA is replicated. And the first thing to look at is that if you look at the two parental strands, notice that the two parental strands, 5' to 3', and the adjacent strand, remember, is going to be anti-parallel going from 3' to 5'. And so as DNA replication occurs, remember that helicase, that enzyme, is going to come in and it's going to separate those two strands of parental DNA. Now, as the replication fork, notice that replication fork is going to the left, so it's opening up to the left. And notice that if you look at the template strand, the top template strand, 3' is over here, which means that the new strand, which needs to be anti-parallel, this is going to be 5', right? Because 3 and 5 go together, 5', and then down here is going to be 3'. Remember that DNA polymerase only goes 5' to 3'. And so this strand, which is going into the replication fork, we call this the leading strand. And the leading strand is made in one continuous motion. Notice what happens on the opposite strand though. The other template, notice here's 3', here's 5', which means that the new strand is going to be 3' here and 5' up here. Remember that DNA polymerase only goes 5' to 3', which means it's going away from the replication fork. Notice it's going down and not going into where it's being replicated. And so when DNA replicates on this strand has to be made in these little fragments. And those fragments are called Okazaki fragments. And these are named after the person who discovered it. Now, in addition, because they're made in little pieces, this strand, this new strand, is called the lagging strand. And so when the lagging strand is made, it's made in short little segments. And what you'll notice is that as this replication fork begins to open up, DNA polymerase is going to go 5' to 3'. When it reaches the previous Okazaki fragment, DNA polymerase is going to fall off the DNA and it's going to go back up into the opening of the replication fork and it's going to go 5' to 3'. And then it's going to go back up and go 5 to 3. And it's going to go back up and go 5 to 3. And what happens is these fragments then need to be sealed. And that's the role of DNA ligase. DNA ligase is going to come in and it's going to ligate or put those two strands together. And so this is just at a very basic level of how DNA replicates. So this is going to be part 2 of our recording, and we finished talking about DNA replication, and in part 2, we're going to start to talk about transcription. So, DNA can encode the information for a huge number of proteins used by living things because the sequence of bases along DNA's handrails can be laid out in an extremely varied manner. And for a long time, scientists always thought that protein is probably the genetic information, because if you think about it, proteins, there were 20 different amino acids, whereas DNA, there were only 4 types of bases. However, many experiments showed that in fact DNA is the genetic information, and the sequence of bases along the strand of DNA are used to code for particular proteins. If you change the sequence of DNA, it's going to change and code for a new protein. And so, depending on what the DNA sequence looks like, it's going to code for a particular protein. Now, we talked about genetics, and we talked about genotype and phenotype. And remember that we said that the genotype is the genes that an organism has. And so the genotype is encoded in its sequence of bases. So what nucleotides in a row. Now, the phenotype, remember, is the physical consequence of a particular genotype. And basically what that means is that the phenotype is a result of what proteins are being produced. And so if you look at the central dogma of biology, we start with DNA. DNA, remember, is the genetic information. It's what's passed from your parents to you. And when you started, remember that you were a single cell. And that one single cell divided into 2. And 2 divided into 4. And so DNA is able to replicate itself. Every time a cell divides, the DNA can replicate. Now, what makes DNA so important, however, is that that information that's encoded in the DNA is used to ultimately code for a particular protein. And the way that that works is that DNA codes for RNA, and specifically mRNA in this example. And notice DNA and RNA, these are both nucleic acids. So we're going to transcribe a similar language. And so this process is referred to as transcription. We're going from nucleic acid language to nucleic acid language. Translation, on the other hand, is where we go from mRNA, and that mRNA is going to code for proteins. Notice this is translating, right? We're going to go from RNA, which is a nucleic acid, to proteins, which are proteins, right? And it's made up of amino acid language. And so we're translating, similar like if you were translating, let's say, from English to Spanish. And so we're going to go from one language to another. And so in this way, DNA is used to code for proteins. And so genotype, the DNA, is going to influence phenotype, which are the proteins that are produced. Now, a genome is all of the cell's genetic information. And a eukaryotic genome is broken up into chromosomes. And remember that each chromosome is one long DNA molecule. So if you look at your cells, your somatic cells, right, all the cells in your body that are not used for reproduction, your somatic cells have 46 DNA molecules, right, 46 chromosomes, and each one is one long DNA molecule. Now, if you look at a particular chromosome, along the chromosome are what are called genes. And a gene is a specific sequence of nucleotides that codes for a protein or an mRNA molecule. Okay, so it's going to be the coding portion of the DNA. And only a small percentage of the human genome actually codes for proteins, only about 1.5%. So if you look along a chromosome, you're going to see these genes that are spread out and spaced along a eukaryotic chromosome. Now, the flow of information is going to be DNA codes for protein. And so there's a little analogy to help you kind of think about how this works. So let's say that my genome is like a collection of all of my cookbooks, right? So all of the cookbooks that I own have recipes in them, and they give me instructions for how to make certain recipes. And my recipes, right, the food that I actually am able to make would be like my protein. And so my cookbooks code for what food I make. Now, if you look at one cookbook, okay, one cookbook could be like a chromosome with multiple genes on it. And so, right, if you think of a chromosome, each chromosome has multiple genes laid out in it. And each recipe in that cookbook is like a gene. Now, let's say that one of my friends wants my recipe for brownies. And so I go to my cookbook and I copy down my recipe for my brownies, right? So I'm reading the information in my cookbook and I'm transcribing it and writing it down on a separate piece of paper. And so that new recipe that I've copied, that's going to be like our RNA, right? So here's our RNA copy of one of the genes that's in this cookbook. Now, I give that recipe to my friend, and that recipe has information in it which tells my friend what ingredients to add and in what amounts. And so the ingredients in this analogy, those are our amino acids, right? We're going to read the mRNA, and the mRNA is going to tell us what amino acids to put in. And so we put in the amino acids by looking at the recipe, and what we get is our final product, which is our proteins. Now, if you think about it, let's say that I made a mistake, and when I was transcribing my recipe, instead of writing, let's say 1 tablespoon of baking soda, instead of saying 1 tablespoon, maybe I make a mistake and I write 1 cup. Now, for any of you that have cooked, you know that most recipes for baking don't require much baking soda. So if I write it down incorrectly, right, if I make an error, my friend is going to read the recipe, right, so it's like reading the mRNA, and she's going to put in the wrong amino acids, right? She's going to put in too much baking soda. And so if you think about it, is that going to affect the taste of her brownies? Absolutely, right? Her proteins are not going to come out properly because she had the wrong information to put in the wrong amounts of amino acids. And so that we're going to talk about in one of the later lectures when we talk about mutations. Okay, but so this is just another analogy to kind of describe the flow of information in a cell. Now, the words of the DNA language are triplet bases that are called codons. And the codons in a gene specify the amino acid sequence of a polypeptide. So remember that we said that if we do DNA replication, in DNA replication, if we look at a strand of DNA, both strands in replication serve as the template. In transcription, both strands do not serve as the template. Only one strand is the template. And so this particular strand, this is our template strand. The other strand, okay, the strand down beneath it, is not the template strand, but is instead what we call the coding strand. And we'll talk about that in just a minute. Okay, so you have one strand that's going to be your template, one strand that's going to be your coding strand. So when we go to do transcription, we're going to remember transcribe from DNA into mRNA. And so an enzyme called RNA polymerase polymerizes or synthesizes RNA, and it's going to read our template, right? So it's going to read an A, and it's going to make a U. Now, notice it's not a T, and remember that that's because RNA doesn't use thymine, instead it uses uracil. And so the enzyme's going to read the A, it's going to put in a U. It's going to read the C, it's going to put in the G. It's going to read the C, it's going to put in the G, and so on down the strand. Now, if you notice, if you look at this mRNA, and you look at this strand here, which we call the coding strand, notice what you see when you look at those two strands. What do you notice that they have in common? And if you think about it, right, notice that this is the exact same sequence as the coding strand, except anywhere there's a T, there's now a U, right? TGG, this is now UGG. And there's 3 T's, There's 3 U's. And so this is the coding strand. It's the same sequence as what is coding, which is the mRNA. Now, why is it 3 nucleotides per codon? Remember that we said that there are 20 amino acids. So if you think about it, if it was only 1 nucleotide per codon, meaning A gave you 1 amino acid, T gave you another, G gave you another, and C gave you another, 4 bases to the first power, meaning that every nucleotide gives its own codon, there would only be 4 possible amino acids. But we know that's not true, right? We know that there are 20. If it were 2 nucleotides per codon, like AA gives us one thing, AT gives us another, 4 to the second, or 16 amino acids would be possible. But again, there are 20. If it was 3 nucleotides per codon, 4 to the third power, 64 amino acids. And so that is enough coverage to give us all 20 amino acids. And so in a minute, we'll talk about why there are 64 possible amino acids, but there are only actually 20. And so what you should look at now is that if you look at these codons, these groups of 3 nucleotides, those are going to code for a particular amino acid. That's translation, right? We're going to translate. We're going to go from mRNA language and translate into amino acid language. And so these codons will be read, and the appropriate amino acid will be brought in. Now, this diagram here on the right is the genetic code. It's basically a readout of the codons on the mRNA and what amino acid they code for. The genetic code was determined in the 1960s, and a group of scientists started out using a translation machinery in a test tube. And so they mixed a UUUU, so this long poly-U sequence, this UUU mRNA, and they mix it with all the components for translation. So they give it the ribosome, and they give it amino acids. And what they found was that if they did this, if they mixed this mRNA with all of the translation machinery, what they got was a sequence, an amino acid sequence. Notice that UUU codes for phenylalanine. And so when they did this, they got an amino acid sequence that was repeating phenylalanine over and over again. And so that told them that UUU codes for phenylalanine. And so they kept doing this with many, many different sequences until they were able to determine the genetic code. Now, if you look at this, the way that you read this, over here on the left, first mRNA base. So let's say we started with the sequence. Let's say our codon was CGG. Okay, so let's say we're trying to look for CGG. So here's C. Second base is here, G. G. So CGG would give us arginine. Now, if you look at this genetic code, there's a couple features about it that will become apparent. And the first is, you notice that there's one in green, AUG. AUG is what we call a start codon, meaning that that's where translation is going to begin. And you also notice it also says MET or start. MET is from the amino acid methionine. So if you're looking at the mRNA, the first AUG, that AUG is going to serve as the start of translation. And it's also going to bring with it the first amino acid, which is going to be methionine. So all proteins initially begin with the amino acid methionine. Now, any subsequent AUG is not going to serve as a start codon. It's instead going to just be another methionine amino acid. The other important thing to notice that there are 3 that are in red, and those 3 that are in red say stop. We call those our stop codons. Those are going to signal the end of translation, and we're going to stop making the protein at that point. Now, I'm not going to expect you to memorize these 3 stop codons, and in fact, you won't be asked to memorize any of the genetic code. If I ask questions related to this, I will in fact give you the genetic code. So, if you look at this, there's a few other features that we need to refer to. And the first is that the genetic code shows something called redundancy. And that is that multiple codons can give you the same amino acid. Notice here, for example, C, U, and any third nucleotide gives you leucine. U, U, A, or G also gives you leucine, right? And so multiple codons can give you the same amino acid. So because of that, we say that the genetic code is redundant. Multiple codons are going to give us the same amino acid. And so remember, there are 64 possible amino acids, but there are only 20 actual amino acids, and that's because the genetic code is redundant. The other thing about the genetic code is that the genetic code is unambiguous, meaning that any codon for one amino acid never codes for another amino acid. So it's unambiguous. It always will give you the same amino acid, like UUU will always give you phenylalanine. Another feature of the genetic code is that it doesn't contain spacers or punctuation, meaning that codons are directly next to one another with no gaps in between. And then lastly, the genetic code is nearly universal, meaning that the same genetic code is used for bacteria, for humans, mice, worms. Okay, and so this genetic code, is universal across many, many organisms. So here's just an example showing you that in fact the genetic code is universal. These mice here, these ones that are green, they're actually glowing green, and they have a gene for jellyfish in them. And this gene that jellyfish has is called GFP, which stands for green fluorescent protein. And in jellyfish, they can make this GFP protein and they fluoresce green. And so what these scientists did was they took the gene for GFP, this jellyfish gene, and they put it into mice, and you can see that the mice glow green, meaning that the mice were able to take that jellyfish gene and make the GFP jellyfish protein. And so this is just an example to demonstrate that the genetic code is nearly universal, that you can take a gene from one organism and put it into another, and it's going to make the right protein. This little mouse here, this is a mouse that doesn't have the GFP gene, and you can see he's not glowing green. So this shows you that this is in fact due to the fact that they have this other animal gene. So this is an example, and this is going to help you with lab. Okay, so in a minute, you're going to go ahead and pause, and I want you to work on this and see if you can go through this. And so what I gave you is I gave you the coding DNA strand. Okay, and so you need to go in and fill out what would the other DNA strand be. So like, what would the complementary DNA strand look like? What would the mRNA codon look like? What would the protein sequence look like? And when looking for the mRNA, keep in mind about what you need to look for to start making your protein. So keep that in mind when you're trying to determine your protein sequence. What does it need to start with? And so go ahead, take a minute, pause, and then you're going to go through and answer this. Okay, you ready? So let's walk through this. I'm not going to write on the slide because unfortunately the way that the video works is that the answer is going to be displayed on the slide at all times. If I write on it, it doesn't add it as I'm writing it. So we're just going to have to walk through this, and you're going to have to write it down as I'm saying it. So if this is a G, it's going to put in C, right? If this is an A, the other DNA strand is going to be T. T is going to be A, G is going to be C, C is going to pair with a G, T is going to pair with A is going to pair with T, C pairs with G, C pairs with G, T pairs with A. Okay, so there's your double-stranded DNA molecule. Now, remember that for protein synthesis, only one strand is going to serve as the template to make the mRNA. And the template is not the coding strand, it's actually the other strand. And so there's two ways for you to approach this, right? You could either read the other DNA, so like if this is a C, that means that this is a G. If this is a T here, right, it's going to be an A. If this is a T here, that means an A here, right? So if this is an A, it's going to pair with a U. Notice what you're starting to have as you go down this mRNA. You'll notice that the mRNA is exactly the same as the coding strand. Remember that the only difference is wherever there's a T, you're going to put a U. So like this would be G, A, U, G, C, U, A, C, C, U. Okay, so now you'll have your mRNA, and then you're going to look for your codons. And remember that translation always starts with an AUG. So here is your AUG. And so this little grouping here, that's going to be your start codon, and that's going to give you methionine. Okay, so there's our start, and then we're going to look at this 3 here. So that's C, U, A. And so we're going to look on our genetic code. C, U, A is going to give us leucine. Okay, so it's going to be methionine, leucine, and then C, C, U. So C, C, U, it's going to be proline. And so we're going to have methionine, leucine, proline would be our protein sequence. Okay, and so in lab this week, you're going to practice a lot of these and just get really familiar with how protein synthesis works. And so now we're going to talk about how transcription occurs. How do we go from DNA to RNA? Now, the first thing to consider is where does transcription and translation occur in the cell? And if you remember back to when we talked about prokaryotic cells and eukaryotic cells, prokaryotic cells like bacteria, remember, lack a nucleus. So in prokaryotic cells like bacteria, the DNA is not contained in a membrane-bound nucleus. Instead, it's in the cytoplasm. And so in this case, transcription and translation are coupled. They're not spatially separated. And so as bacteria is doing transcription, so as it's making its mRNA, simultaneously it's going to translate it. Okay, it's going to read the mRNA and synthesize the appropriate amino acid sequence. And so these two things can happen at the same time. In eukaryotic cells, for example, like ourselves, remember that eukaryotic cells have a nucleus. And the nucleus is made up of a nuclear envelope, so it's a membrane-bound structure. And inside the nucleus is the DNA, right? And so the DNA is in the nucleus, but the ribosome, which does translation, is actually in the cytoplasm. And so you'll notice transcription and translation are spatially separated. In eukaryotic cells, transcription happens in the nucleus because that's where the DNA is. But translation occurs in the cytoplasm because that's where the ribosome is. And the ribosome, remember, is our organelle responsible for protein synthesis. And so what this allows then is that RNA is actually processed before it leaves the nuclear pore. And so we'll talk more about RNA processing in the next lecture. So when talking about transcription, there are 3 stages. And when we get to translation, you'll notice they're the same 3 stages. We have initiation, elongation, and termination. And it's kind of nice because the names of the stages actually tell you what they do, right? So the first stage is going to be our initiation. And this is where transcription is initiated, or basically begins. And so in initiation, if you look at a gene, at the beginning of a gene is a sequence called the promoter. And a promoter is a very specific DNA sequence that the enzyme RNA polymerase is going to polymerize or make the mRNA. And so RNA polymerase is going to bind to that DNA at the promoter, And once it binds, it's going to initiate transcription. Okay, and so initiation involves RNA polymerase binding to the promoter to begin transcription. Now, remember that when you look at a DNA, only one strand is going to serve as the template. The other strand is the coding strand. And so in this particular example, this is our template strand. Notice here's 3, here's 5, which means on the mRNA, this is 5, this is 3. And just like DNA polymerase, RNA polymerase only goes 5' to 3'. Okay, it has to add on to the 3' end. And so RNA polymerase is going to separate the two strands of DNA, and we're going to start to do elongation. It's going to read the DNA sequence, and it's going to put in the appropriate mRNA. Okay, so it's going to do complementary base pairing. And so it's going to read the DNA, and it's going to synthesize its mRNA. That's the elongation phase. Termination is where we're going to terminate, right? That's when a transcription ends, and we'll talk about why in just a minute. So a little bit more about initiation. If you look in the promoter of a eukaryotic cell, remember that the promoter is where RNA polymerase binds to initiate or turn on transcription. And in the promoter is a sequence called a TATA box. Okay, T-A-T-A, we call it a TATA box. And a TATA box, notice the sequence T-A-T-A, and then there's a couple more A's in there. This TATA box allows proteins called transcription factors to bind. And so transcription factors are basically just DNA binding proteins that recognize certain sequences, and they, in some instances, help recruit RNA polymerase to the promoter, meaning that they help RNA polymerase bind to the promoter so that it can initiate transcription. And so these proteins help bring RNA polymerase to the promoter, and they help initiate transcription. And so this is going to form something called the transcription initiation complex. Okay, it's this group of proteins that helps RNA polymerase bind to begin transcription. And so again, we're going to do elongation. RNA polymerase is going to separate the two strands of DNA. It's going to read the template, and it's going to put in the appropriate nucleotide complementary base pair. Right? So if it's a T, it's going to put in an A. If it's a T, it's going to put in an A. If it's an A, it's going to put in a U, and so on. And so the new mRNA is going to be made by adding on at the 3' end free RNA nucleotides. This is going to continue until RNA polymerase reaches a sequence called the terminator. And when it reaches that terminator, it's going to terminate or finish basically transcription. And RNA polymerase at the sequence is then going to fall off, and transcription is complete. And so those are the steps in transcription. So here is a question, and again, you should pause it, think about it, and then answer. And the question says, transcription, is it red, occurs on the ribosome? Yellow is the final process in the assembly of a protein. Green is the synthesis of RNA from a DNA template. Blue is catalyzed by DNA polymerase. Purple occurs in the cytoplasm of eukaryotic cells. And now what I want you to do when thinking about this as well is not only find the right answer, but also convince yourself why the other answers are wrong. Okay, so go ahead, pause the video, and then when you're ready, replay it, turn it back on again, and listen for the answer. So go ahead, pause the video. So if you said green, you are correct. Transcription is the synthesis of RNA from a DNA template. Now let's go through why the other answers are not right. Transcription occurs on the ribosome. That's not true because translation is going to occur at the ribosome. Transcription, if you're talking about eukaryotic cell, transcription takes place in the nucleus, whereas in bacteria, transcription takes place in the cytoplasm, but it doesn't occur on the ribosome. It's not yellow, is the final process in the assembly of a protein, because this happens before the protein is even being produced. It's not blue, it's catalyzed by DNA polymerase, because DNA polymerase makes DNA. RNA polymerase is going to be the one responsible for transcription. RNA polymerase is going to make RNA. And then it's not purple, occurs in the cytoplasm of eukaryotic cells. Now, if I hadn't said eukaryotic cells, transcription could occur in the cytoplasm, right? Prokaryotic cells, transcription occurs in the cytoplasm. But in eukaryotic cells, transcription occurs in the nucleus. And so that is why purple is incorrect. So this is going to be part 3 of your video. And in this video, we are going to move on and talk about translation. So we just finished talking about transcription and how DNA is used to make an mRNA. And in this lecture, we're going to talk about how mRNA is then used to code for protein. And so I have a question for you. And once I read it, go ahead and pause it, think of your answer, and then when you're ready, go ahead and push play again. And so the question says, an organism's genetic information is stored within the sequence of blank. This information is transcribed into a sequence of blank, which are then translated into a sequence of blank. Is it red, DNA bases, amino acids, RNA bases? Yellow, RNA bases, DNA bases, amino acids. Green, amino acids, DNA bases, RNA bases. Or blue, DNA bases, RNA bases, amino acids. So go ahead and pause. Okay, now, did you say that the answer is blue? If you did, then you're correct. Okay, and so the information is stored within DNA, which is transcribed into RNA bases, which is then translated into amino acids. And so this blue is the correct answer. Now, remember that we said that for eukaryotic cells, organisms that have a nucleus, transcription and translation are spatially separated. In the nucleus, remember in eukaryotic cells, that's where you're going to find the DNA, and that in the cytoplasm is where translation is going to occur. And we mentioned that because they're spatially separated, RNA actually undergoes processing before it exits the nucleus. And so what does that really involve? One of the things that happens during RNA processing is that a 5' cap is added and a 3' tail is added. And the cap and tail serve a variety of functions. They help with the stability of the mRNA. They also help with starting and initiating translation. And another sequence that you should be familiar with is that if you look here on the DNA, here's the exon, here's the intron. Now what are those? Exons are the express sequences. These are the ones that actually code for the protein. Introns, on the other hand, these are called the intervening sequences. These are the sequences that need to be cut out, and they do not code for the protein. And so when you look at the DNA, you're going to see exon, intron, down the gene. During transcription, RNA polymerase is going to read the entire DNA. It's going to transcribe both the exons and the introns. And so it's going to make this primary transcript. And again, the primary transcript is going to have both the exons and the introns. But the RNA needs to be processed. And so it does something called splicing. And splicing is going to remove the introns. And so a structure called the spliceosome is going to come in. It's going to recognize these junctions between the exons and the introns. And it's going to loop out the intron and put the exons back together. And so now you get your mature mRNA. And your mature mRNA has all the coding sequences directly next to one another. We have now removed all of the introns. And now that mRNA is ready to exit out the nuclear pore and go into the cytoplasm to do translation. And so we're going to talk about how we go from RNA to protein. So, in order to understand translation, we need to talk about another type of RNA. And this type of RNA is called a transfer RNA. And a transfer RNA acts as the interpreter. It's the one that's going to read the mRNA language, it's going to read the codon, and it's going to bring with it the appropriate amino acid. If you look at the structure, here's the structure of a tRNA, and you'll notice that it has something called an anticodon. And what is an anticodon? So let's write something out real quick. So let's say that my DNA sequence is going to be let's say ATC. Okay, so this is just some made up DNA sequence. Let's say that my DNA sequence is ATC. And let's say that this DNA is going to be my template strand. If this is my template strand, that means that my mRNA is going to be complementary. So A is going to pair with U, T is going to pair with A, C is going to pair with G. So this is my mRNA, and that mRNA is going to be your codon. Now, if you look at the tRNA, this means that the tRNA anticodon is going to be complementary to the mRNA codon. So if this is a T, or I'm sorry, if this is a U on tRNA, it's going to be an A. And if on mRNA it's an A, it's going to be a U. And if on mRNA it's going to be a C, the tRNA is going to have, or I'm sorry, if the mRNA has a G, tRNA is going to have a U. And so the anticodon is on the tRNA, and the anticodon is going to be complementary to the mRNA codon. Okay, and so notice U is going to have A is going to have U, G is going to have C. And so this is the way that the tRNA is going to act as the interpreter. It's going to read the mRNA codon, and it has another important site that's called the amino acid attachment site. And this is where the amino acid will bind to. And so in this way, the tRNA is able to bring in the appropriate amino acid based on its anticodon, which recognizes a very specific codon on the mRNA. Now, remember that when we talked about cell organelles, we mentioned that the ribosome is responsible for protein synthesis. And so the ribosome is going to be responsible for translation. Remember back to when we talked about the ribosome, it has two subunits, a small subunit and a large subunit. And each subunit is made up of ribosomal RNA and proteins. And so remember we talked about that the nucleolus did ribosomal RNA synthesis, and it was also responsible for ribosome assembly. And so remember that the ribosomal RNA made the nucleolus, the proteins are going to come into the nucleus, ribosomal RNA is going to get put together in the nucleolus with it, and it's going to make our large and our small subunits. And the large and the small subunits don't come together until translation. And so the nucleolus is only responsible for synthesizing or putting together, assembling the large subunit, and it's also responsible for assembling the small subunit. But it doesn't put them both together until translation. The large and small subunit will come together in the cytoplasm. Now, an important feature of the ribosome is that it has binding sites for both mRNAs and tRNA. And so you could see that it's going to bind the mRNA, and it also is going to be able to bind the tRNA to bring the anticodon with the codon. And notice here you can see that this has two tRNAs bound together side by side. And we'll talk about how translation works and how we can add on an amino acid to a growing polypeptide chain. So, just like transcription, translation has 3 steps: initiation, elongation, termination. So the same 3 steps that transcription has. So let's start by going through what happens in each step. So in initiation, the first thing that's going to happen is that the mRNA binds to a small ribosomal subunit. Okay, so here we see, here's our mRNA, here is our small ribosomal subunit, and the small subunit is going to bind to the mRNA. Now, remember that when we do translation always starts, the start codon is AUG. And so notice that this sequence here, this we call the UTR. UTR stands for untranslated region. Notice there's no AUG. So translation doesn't begin until the first AUG. And that first AUG is going to be bound by a tRNA that has a sequence of UAC, right? It's going to be complementary to the AUG. And it's going to bring with it the amino acid methionine. Remember that all proteins initially will start with methionine as the first amino acid. And so once that tRNA docks at the mRNA, now the large ribosomal subunit will come in. It's going to bind to the small subunit, and now we have initiated translation. We have our functional ribosome, and now we're ready to translate the mRNA sequence. Elongation, right, is going to be where we elongate or grow our polypeptide chain. And so what's going to happen is remember that we said that for this ribosome that there are two spots where the tRNAs can bind. And so the next tRNA is going to come in and it's going to recognize the next codon. And so notice that your next codon here is GGA. And so a tRNA that's complementary that has CCU is going to come in and bind. Now, the important thing that's going to happen then is when these are coming together on the ribosome, those ribosomal RNAs are actually catalytic. They're able to catalyze or speed up the peptide bond between methionine and the next amino acid, so in this case glycine. And so it's going to form that peptide bond between those two amino acids. Methionine is going to leave its tRNA, and it's now going to be on the glycine. That tRNA is now going to leave, and then the glycine is going to shift. Okay, and so the ribosome is like a ratchet. It's now going to shift down to the right, so it's going to go this way. That tRNA is going to leave, so here you can see the tRNA leave. And then now there's a new spot that can be occupied by the next tRNA. And so that next tRNA comes in. It's going to catalyze the peptide bond between these amino acids. Once it does that, the chain is now going to be over here. This tRNA is going to leave, and the next one's going to come in here. And so this is the elongation. This is going to be growing and lengthening that polypeptide chain. And then termination happens when we get to a stop codon. And so when you get to a stop codon, remember that there are 3 of them. The stop codons don't have a tRNA that's going to bind. Instead, they have a protein called the release factor protein. And this release factor protein is going to recognize the stop codon. It's going to bind, and because there's no amino acid to be brought with it, that's going to signal the end of translation. And so once we get this release factor and there's nowhere to add on to the polypeptide chain, now the ribosomal subunits are going to dissociate, they're going to come apart, and your polypeptide is now released and is fully formed. So how fast does translation occur? And the answer to that is that when we do translation, it's not that just one ribosome is going to bind at the beginning of the mRNA and it's going to go all the way through, and then it's going to come back and do it again and again. Instead, many ribosomes bind along the length of an mRNA, and they form something called the polyribosome structure. And so here's an electron micrograph. You can actually see this. So here you have your mRNA, and all of these little like dots, those are going to be your ribosomes. And so you can see that there's many, many ribosomes along the length of an mRNA. And so because of this, because they're going to just kind of move along the mRNA, this is going to allow translation to be very, very fast. E. coli, which is a common bacteria that can be found in your gut. E. coli can string 40 amino acids per second. So an average size protein of 400 amino acids is built in just 10 seconds. Another example that really shows you how fast this is that a human immune cell, so your B cells, can manufacture 2,000 identical antibody proteins per second. So just think about that for a minute, how fast that is. It's like, boom, it just made 2,000 antibody proteins. So the translation is very, very, very rapid. And the reason that your immune system is so efficient is that you have these cells that are called memory cells. And your memory cells recognize a foreign invader that they've already seen. And once they see that, they have that information stored so that if you come in contact with that again, your immune system just cranks out antibodies to fight off that foreign invader. And so this is why you can't get sick with the same thing again and again. For example, right, if you've ever had the chicken pox, you know that once you have the chicken pox, you're not going to get it again. But if you think about the flu, right, you've probably had a flu many, many times. And that's because what ends up happening is the flu virus actually can mutate and it changes its shape. It's like a chameleon. It changes its shape, and so the antibodies that normally would recognize it, because its structure is a little bit different, those antibodies won't recognize it, and the flu is kind of just hiding out, and your body has to figure out new antibodies to produce. And so it's just something to kind of think about how amazing your bodies actually are.
Why?
So now let's look at some of the differences in prokaryotic cells. Remember that for prokaryotic cells like bacteria, that their chromosomes are circular instead of linear like your chromosomes. And so because of this difference, the way that bacteria replicate their DNA is a little bit different. And you can remember that for when we talked about selective toxicity and that some antibiotics target bacterial replication. And so one of those ways in which we target bacterial replication, remember, was by inhibiting DNA gyrase. And so let's talk about how bacteria replicate their DNA. And so again, you can see that here we have the origin of replication. And remember that that is where replication begins. It's a specific DNA sequence that proteins bind to that recognize, and they start to separate the two strands of DNA. Now, just like in eukaryotic cells, at the origin of replication, the replication fork goes in both directions simultaneously. And so what you get is that the structure of DNA, we call this in bacteria, it's called theta replication. And so theta is a Greek symbol, and the reason they call it theta replication is it looks like the Greek symbol for theta. For bacteria, they use DNA gyrase. And the purpose of DNA gyrase is to separate the two circular DNA and relaxes what's called supercoiling. And so DNA gyrase is an enzyme that bacteria use to replicate their chromosomes that eukaryotic cells do not require because eukaryotic cells do not have circular DNA like bacteria do.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Another difference between prokaryotic and eukaryotic cells is how they do transcription and translation. And so you have to remember that for eukaryotic cells, transcription and translation are spatially separated, meaning that transcription, which remember is where we go from DNA to mRNA, transcription in eukaryotic cells happens in the nucleus, while translation, which is going from mRNA to protein, that happens in the cytoplasm where the ribosomes can be found. However, in prokaryotic cells, there's no spatial separation of transcription and translation. Remember that prokaryotic cells don't have a nucleus. And so for prokaryotic cells, transcription and translation happen simultaneously, meaning that as the cell is making the mRNA, simultaneously, it's also going to be translating it. And so remember that one of the differences between prokaryotic cells and eukaryotic cells is that prokaryotic cells don't undergo RNA splicing, meaning that they don't have time to cut out introns because, again, as they're transcribing it, as they're making the mRNA, they're also simultaneously translating it and making the protein.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
And so, question for you. So the question is, transcription, is it red, occurs on the ribosome? Yellow is the final process in the assembly of a protein. Green is the synthesis of RNA from a DNA template. Blue is catalyzed by DNA polymerase. Or purple occurs in the cytoplasm of eukaryotic cells. And so I want you to think about the question, pause the video, and then when you're ready, go ahead and push play to hear the answer. Okay, pause. Okay, so if you said green, you are correct. Transcription is the synthesis of RNA from a DNA template. Remember that DNA codes for RNA, and we call that transcription because we're transcribing a similar language. DNA and RNA are both types of nucleic acids. It's not red, occurs on the ribosome, And that's because transcription doesn't occur on the ribosomes, translation does, right? So translation is where we read the mRNA and we put in the appropriate amino acids. And so translation occurs on the ribosomes, but not transcription. It's not yellow is the final process in the assembly of a protein. That's not true because this happens before the protein is even being made. It's not blue is catalyzed by DNA polymerase because DNA polymerase, remember, is responsible for replication of DNA. Transcription is making RNA, and so transcription is actually catalyzed not by DNA polymerase, but instead RNA polymerase. And then it's not purple. It doesn't occur in the cytoplasm of eukaryotic cells. Notice it had to say eukaryotic cells. And that's because in eukaryotic cells, transcription takes place in the nucleus because that's where the DNA is found. If that had said transcription occurs in the cytoplasm of prokaryotic cells, then purple would have also been true. But because it says eukaryotic cells, that then makes the statement false because transcription doesn't occur in the cytoplasm of eukaryotic cells.